welcome to the n-Lorem podcast series, a series that focuses exclusively on patients now referred to as having nanorare mutations. I'm Stan Crook, and I'm the founder, chairman, and CEO of n-Lorem. n-Lorem is a nonprofit foundation that I initiated in January of 2020. Our mission at n-Lorem is to take advantage of the technology we created at Ionis Pharmaceuticals, Antisense technology, or ASO technology, to discover, develop, and provide experimental ASO treatments to nanorare patients, and to do that for free for life.
For the n-Lorem Foundation, N of 1 doesn't mean alone, because every patient matters. Hope for nanorare patients and their families has never been more within reach. Visit n-Lorem.org today to donate and make hope possible. This episode of the n-Lorem Patient Empowerment Program podcast is proudly sponsored by Hanjin Biotech. With more than 26 years of experience in the nucleic acid industry, Hanjin is a specialized CDMO and raw material supplier committed to providing high-quality and high-value products and services with the best-in-class lead times.
Today I'll be talking about a subject that's actually really quite complex, and I hope I can simplify it and make it understandable for you. We'll be talking about iPSCs. Now, that's a term that I suspect many of you have heard, and many probably wonder what it really actually means. Well, the term iPSCs is an acronym, and it stands for Induced Pluripotent Stem Cells. And so the first step is to figure out what that actually means. So let's take each word. Induced. As you know, in your body, you have two big categories of cells: germ cells and somatic cells. Germ cells are the cells capable of creating a new human. They're sperm cells and ova or egg cells. Somatic cells are the cells that make up your body, and they are derived from germ cells. Somatic cells are not capable of creating new human being, but still have all the genetic information that germ cells have. That is, they have a full genetic reference library, but only use some of the books in that library, and they use just some of the genes, therefore. So an iPSC is a somatic cell that has been induced to become germ cell-like. That means that the induced cells can be grown in the lab and reproduce itself indefinitely, or themselves indefinitely. That means that we can make large quantities of human cells for the first time. Operationally, typically what happens is that blood or skin cells are collected from a patient, and the skin cells are called fibroblasts. So you'll hear fibroblasts, skin cells, blood cells. They're just the beginning cells that are easily accessible. And so we collect those from a patient, and then we transfer those cells to dishes, to tissue culture, and subject them to treatments with chemicals that cause them to reprogram themselves into germ-like cells. In short, they are induced to become germ-like cells. Pluripotent, what does that mean? Germ cells are capable of creating all the types of cells in your body. That ability to cell to create any type of cell is called pluripotency. And so germ cells are pluripotent. Pluri, many, potent. Many different abilities. Big word, simple idea. So an induced pluripotent cell is a somatic cell that has been reprogrammed, induced, to become germ-like. That is, it is pluripotent. It is capable of becoming other kinds of cells and doing all the things that any type of cell might do. That's an amazing new attribute. So what's a stem cell mean? Well, stem cell simply means a cell that's capable of reproducing many more cells of the same type, so it can reproduce itself. So an induced pluripotent stem cell is a somatic cell, typically blood or fibroblasts from your skin, collected from a patient, treated with chemicals so that they become germ cell-like and have the capability of reproducing themselves and then being converted into all kinds of other cells. Okay, so key point. iPSCs stand for somatic cells, typically blood cells or skin fibroblasts, that have been reprogrammed to behave like germ cells. I can't begin to emphasize how fundamental that is. iPSCs are the product of multiple Nobel Prizes over the decades, and the ability to be able to collect human cells from patients and use them as we are using them makes what we do possible. Without iPSCs, n-Lorem does not exist. It's that simple and that important. Okay, so now we need to understand some things that are going to lead us to understand what differentiation is, since we just de-differentiated these differentiated cells. So let's talk about chromatin. As I mentioned many episodes ago, well, I doubt you remember, but if you stretched the DNA in any one of your cells, it would be meters long. Vastly bigger than a microscopic cell. So how in the world do you fit all that DNA in the nucleus of your little tiny microscopic cells? You do it by compressing those files. You can think of it like compressing a file so you can send a PDF. And you do that by putting your DNA into a structure called chromatin. And chromatin is designed to compress all that chemical information into a much smaller space. That's what chromatin is. Chromatin comes in two architectural varieties, super compressed and relaxed. Sometimes compressed is called closed, and sometimes relaxed is called open. Sometimes compressed is called euchromatin, and relaxed is called heterochromatin. It really doesn't matter. Those are just all different terms meaning two architectural shapes of chromatin. One that's extremely condensed, very closed, and one that's more relaxed, but they're still highly condensed. Okay, so most chromatin is super condensed. Now, as you can imagine, if you take all the genes in a bunch of your chromosomes and condense them into this tiny little package inside your nucleus of condensed chromatin, they might not be very accessible to all the machinery that you need to use your genes. That's exactly right. So everything's simple. It's as simple as taking your house and compressing it into a tiny little ball. You're not going to be able to use your living room if you can't get in the ball. So that's what supercompressed chromatin is. And that means that the genes in supercompressed chromatin are silent. They cannot be used. And when it's more relaxed is the time when you can use the genes that you have. Now, as you can imagine, something as important as what genes you use and what genes you don't, something as important as chromatin, how you compress all that genetic information into the nucleus of your cell, that's got to be really tightly regulated. And a lot of different systems in your cells that manage the shape and architecture of your chromatin. Some of it involves chemically altering DNA. Some of it involves chemically altering proteins like histones and other kinds of proteins. All these interactions are integrated, orchestrated, and stunningly complex. And they manage the shape of your chromatin with the ultimate goal of defining what genes are actually being used at any given time. I could get into all of that goes on with chromatin, but you don't need to know it to understand what iPSCs are and why they're important. So we're just going to focus on what you need to know. If you have questions, I'm happy to walk you through, what those of us in biological science spend a lifetime learning about. So key point, the DNA in your cells is compressed into a structure called chromatin. Chromatin exists in two forms, supercondensed or relaxed. Only the chromatin in relaxed configuration can be used to transcribe your genes, to use the genes that are available. So that brings us then to why you have chromatin and why you use only some of your genes and not all of them. Well, and that's called differentiation. We've been talking about de-differentiating somatic cells into germ-like cells. We're now talking about the forward process. And so differentiation is the process that creates all the types of cells that make you up from a single fertilized egg cell. And so that fertilized egg cell is kind of an amazing thing when you think about it. You've got one tiny cell that can expand to create the body that you are and all the different functions that you have to engage in every minute of every day to be alive and to regenerate, to stay alive, to respond to stress, to deal with all the things that life brings you. It's astonishing that all that information is packed into one fertilized egg cell that is in your mother's uterus, and you start that way. That process of taking that single cell and creating you is called differentiation. So since the first step in using a gene is to convert the information in that gene to RNA, and remember that process is called transcription because it's like Xeroxing, right? Same language as in DNA goes in RNA. So that process is called transcription. So if it makes sense that if you want to control what genes are actually used, you will control transcription. And you do that through a set of proteins called transcription factors and other systems and proteins that convert the architecture of chromatin to compressed or relaxed. Those two giant processes, enormously complex processes, are the processes that allow you to become you from one fertilized egg cell. Okay, so you have many proteins that bind to DNA, and they bind to specific sequences in DNA. Those proteins are called transcription factors. Not only do you have many, many different kinds of transcription factors, you have some that are master regulators. These are transcription factors that turn on or off whole families of genes. So you have transcription factors that control individual genes and individual groups of genes, and you have master transcription factors that shut entire sections of your genetic library off. Close the door. Can't be used. For all purposes, basically no longer exist. Those are the master transcription factors. So differentiation, therefore, is the process of selecting just a tiny fraction of the genes that are available to be used in the cell at any given time. Controlling transcription is the key to differentiation. So key point, differentiation is the process of control of what genes are transcribed. Transcription is controlled by transcription factors and processes that alter chromatin architecture. Another key point, iPSCs are somatic cells that are caused to go through the reverse process, de-differentiate, and return to a germ-like state, like a fertilized egg cell, ready to make any cell you want. Okay, so why are iPSCs important? Well, iPSCs are important because for the very first time in medical history, we can collect cells from patients and induce them to become germ-like. That allows us to grow large quantities of these cells, and then we can reverse the process and differentiate them into somatic cells of interest that we want to study, like muscle cells, heart cells, eye cells, brain cells, and so on. And we can differentiate them into very specific types. So in the brain, you've got neurons of various kinds. You've got glial cells that include astrocytes and dendrocytes and all kinds of cells that sort of work along with neurons. And we can differentiate a germ cell-like cell, an iPSC, into specific types of cells that populate the CNS or populate other organs. That's really extraordinarily powerful. And we do that by adding back transcription factors to cells in culture and allowing them to change over time. And the process really boils down to turning off a bunch of genes and turning on a select few that make a cell a particular type of cell. Without the ability to create iPSCs and then redifferentiate them, we simply could not treat nanorare patients today. It would not be possible. So while iPSCs can be de-differentiated into nearly any type of somatic cell, like skeletal muscles, liver cells, and whatnot, neurons, that is central nervous system cells that store and propagate information, are the most common cells that we use because most of our patients have neurological diseases. But we can create whatever cell type we like. Okay. And obviously, because blood is easy to get and we can do a little biopsy of skin, we can access a cell type converted to a de-differentiated germ-like cell, iPSCs, and then use that to recreate any cell type we like. So there's nothing magic about having you come in and have your blood drawn or a biopsy of your skin. It's just these are cells that are accessible easily to us and are not traumatic to you for us to collect. In contrast, if we wanted brain cells or heart cells, that tends to be a little challenging. So making iPSCs and redifferentiating them into cells that we like to study is very time-consuming and very expensive. And one of the things that's working right now is we're working very hard to make that process less expensive and less time-consuming. We've made a lot of progress in that, but there are many, many laboratories around the world working on this. So the process to create the cells that we study actually takes months. That is why it's essential very early in the process that we collect your blood cells or your skin cells so we can get to work making the cells we need to study. That's just lost time. And those months are times while your disease is progressing, and that just makes you harder to treat. And so anything that we can do to reduce that time, collect your cells early, or shorten the time that it takes to make those cells is going to be really helpful. And we're working hard on all kinds of ways to do that, and we've made good progress. And anything that we can do to make it less expensive will save us quite a bit of money, because we have 100 and maybe 200 different human cell lines now that have to be maintained, grown, and kept de-differentiated. And all of those chemicals are tremendously expensive. So that's why iPS cells are important. So iPSCs represent an enormous scientific advance, and that could not have happened without decades of basic research. And as I mentioned, the research that I just summarized is the product of so many great scientists working for so many decades and so many Nobel Prizes and other prizes awarded for it. I can't emphasize enough that science is only powerful as it accumulates. And anything that disrupts the progress in science can set science back and medicine back decades. So it's really important that each and every one of you support the scientific process and the funding of scientific process and the independence of scientific process. Meddling by politicians in science harms patients for decades. We cannot allow that to continue. Anything that interferes with the progress of science has profound consequences for decades. So iPSCs are vital. We can do it. We are doing it every day. We're working very hard to reduce the time and cost that iPSCs represent, and they reflect centuries of advances in biological sciences. That could only happen because of the continuum of science and the joy and the privilege of being curious about how we work and why we break down and what we can do when a disease happens. Without that process, without that curiosity, without that commitment, without that time course, without centuries of effort, uninterrupted until very recently, none of this would be happening. There would be no hope for nanorare patients. So we need every person who cares, armed and voting for people who understand science and will treat science respectfully for the value that science creates.
n-Lorem is a nonprofit committed to discovering and providing personalized experimental treatments for free, for life, to patients with genetic diseases that affect 1 to 30 patients worldwide, referred to by n-Lorem as nano-rare. Many of these patients progress and die without ever achieving a diagnosis. This is where n-Lorem comes in. They do the impossible by providing hope, and for those that they can help, free lifetime treatment. For more information about n-Lorem or today's episode, visit n-Lorem.org. Any questions can be sent into podcast@n-Lorem.org. Search n-Lorem on Twitter, Instagram, YouTube, LinkedIn, and Facebook to connect with us. Please rate and review the podcast on Apple, Spotify, or wherever you listen. This truly helps us climb the charts and allows others to find the show. This podcast is hosted by Dr. Stan Crook. Our videographer is John Magnuson of Mighty One Productions. Our producers are John Magnuson and Kira Deneen of DNA Today. Thank you for listening.